Today we will have three presenters, myself as well as Antoine Jacot and Michael Garcia, both from Poietis. We will each do a quick introduction of ourselves before our section within the webinar. Today's session is the first of three in a webinar series, marking the start of our partnership between Synteca Instrumentation and Poietis. During the webinar, I'll take a few minutes to make the introductions to both companies, and then we'll introduce you to the NGB-R system for 4D bioprinting. Antoine will then review some of the basics of bioprinting and introduce you to more detail about the NGB-R system and various approaches that are used within the system. Finally, Michael will join us to talk about the specific application of using the NGB-R system. Before we begin, I'd like to briefly mention a few housekeeping items. We do anticipate today's presentation will be about 45 to 50 minutes. This should leave us with some time at the end to answer some of your questions. Please submit the questions in the Q&A dialog box at the bottom of your screen throughout the presentation, and I'll take time at the end to work through as many questions as we can with the team in the hour that we do have together. We will create a transcript of the questions we do answer, as well as those that we are unable to answer during the session, and we'll send that out to everyone within the next couple of days. Please also note that we are recording today's webinar session, so if for some reason you lose connection or you can't hear everything clearly, you will have access to the video file later. And now, without further ado, let's get things started. As I mentioned in the introduction, I will be one of the presenters for the first part of the webinar today, along with Antoine and Michael, who are both from Poietis. I manage the cellular and imaging divisions here at Synteca Instrumentation, where we have a whole portfolio of different products related to both in vitro as well as in vivo research, with many different application areas that you could think of. Today, we hope to introduce you to the newest addition to our product portfolio, and that is the NGB-R system from Poietis, a disruptive 4D bioprinting system for fabricating complex, functional, and of course, living tissues. During today's webinar, I'll start by reviewing the new partnership between Synteca and Poietis, talking a bit about each of the companies and the goals behind our partnership. We'll then take a brief pause and run an audience poll so that we can learn a little bit more about the work that you're doing in your labs. I'll then continue to introduce the NGB-R system, and Antoine will then jump in to discuss a little bit more detail about the idea behind the description of the NGB-R being a disruptive technology, as well as the cytocentric approach to bioprinting, which is at the core of the NGB-R system. Michael will then take over and talk about the advantages of advanced cell printing or cell patterning and the 4D bioprinting approach. And he'll do this by highlighting a specific example. I'll then rejoin the conversation by running a final audience poll, and we'll finish up with a question and answer period. As I mentioned earlier, please feel free to enter any questions throughout the session in the Q&A dialog box, and we'll get to them towards the end of the presentation. So to get things started, many of you have seen the announcement about the new partnership between Synteca and Poietis, and that happened about a month ago. As a bit of background, Synteca was founded in 2017, and we have offices in London, Ontario, Canada, as well as in the US and Europe. Synteca was formed and has a mission to identify new and innovative systems while providing scientific support to connect researchers with well-designed equipment. We work hard to identify and evaluate each piece of equipment that we represent, ensuring that it has a need within the research community and that it was well designed and manufactured, providing us with confidence that the researchers will see the value and want to include these systems in their labs. Synteca is the exclusive distributor of the NGB-R system within North America. Poietis, on the other hand, is the manufacturer of the NGB-R system and was founded in 2014 in Bordeaux, France. The company was formed with several goals in mind. The most relevant to the discussion today is the mission as it relates to translational research, that is to provide equipment that will allow researchers to design and make actual functional tissue and not just 3D scaffolds. Additionally, in terms of regenerative medicine, the team at Poietis wanted to help researchers bring new and effective tissue engineering therapies to patients. We are all really excited to bring the system to you today and explore the possibilities of how the system could help you in your research. I'd like to start now with our first poll. So it'd be great to learn a little bit more about the work that you're doing. in the bioprinting research area. So perhaps are you already using bioprinting, either using extrusion techniques or inkjet techniques or even laser assisted techniques in your work? Please feel free to choose as many as you want within both the first and second questions. Secondly, it would be great to get a bit more of an understanding about the work that you are doing. Are you working in basic science? Are you taking a look at biomaterials and bioengineering? Or are you working in a slightly different area in terms of cosmetic and in vitro testing? Again, please feel free to select all that you'd like within either question. I'll give everyone just a couple more seconds to finish up their responses. And it looks like we have some people doing some bioprinting already and some that are looking to add it into the future, and really a great representation across all of the different areas, including bioengineering, translational regenerative medicine, as well as cosmetics and in vitro testing. Great, thank you for taking the time to answer the poll questions for us. I'll now move on to a little bit of an introduction about the NGB-R system. So what does NGB-R stand for? I've used that acronym many times as we started the webinar today, and it stands for Next Generation Bioprinting, and the R stands for research. And that's because Poietis is in the process of a second system, which would be more for clinical applications. The NGB-R system has been designed as a standalone, fully integrated bioprinting platform. You can see in the video that the system includes a biosafety level 2 cabinet. This is included to allow you to be able to perform not only the basic fabrication activities, but also to prepare the cells and the cell culture media that you would need and the bioinks that you would need to perform your printing. As we walk through this section, you will see the system includes high-end robotics as well as advanced automation tools, providing high precision and reproducible results. To the right of the system, when we get through the video, it'll loop around again. You can see that there's an onboard computer, which allows the user to fully integrate all of their different steps that they want to perform within the fabrication process. As I mentioned in the previous slide, the system does include a biosafety level 2 cabinet, providing workspace and area for you to prepare the cells as well as your bioinks when you're getting ready to print. There is a 6-axis robotic arm on the system. We'll see that in action in a few slides from now. This is an industrial grade arm, which helps provide the precision necessary to perform the intricate patterns which may be necessary to fabricate the functional tissues that you'll see later. There is a touchscreen interface on the system to allow the users to take their cell patterns they designed on the CAD software and to then program the biofabrication process. There are different techniques used on the NGB-R system for bioprinting. This area here has the option for up to 3 bioextrusion or microvalve heads. And then there is the laser assisted head as well integrated into the system. The laser assisted technique allows us to get single cell resolution, as well as to provide the ability to have those intricate patterns that we'll talk about in a few slides from now. To ensure the precision of the instrument, there is actually also a built-in microscope, which can take images layer by layer as the fabrication process is proceeding to ensure that the resolution and accuracy is there for where the cells have been printed. The software suite of the NGB-R system has been designed to allow users to easily design and plan their cell patterns within the CAD software. These layers and patterns are then moved over to the integrated computer on the system, which you saw a few slides ago, to allow the user to fully plan their fabrication steps, including layer by layer integration to complete the fabrication. Along the way, the user may want to add in images from that integrated microscope that we saw on the slide before to ensure that the fabrication steps are accurate and we can watch as the fabrication progresses over time. As more is understood about the 4D bioprinting and the cell patterns that may be required to achieve your specific end goals, the intuitive planning tools within the NGB-R software will be even more important to the work that you're doing, allowing you to create customized layers and then to fully plan the intricate details of your biofabrication process. Antoine will touch on this concept of cytocentric bioprinting in his section. However, I wanted to take a few minutes here just to introduce the concept as it sits at the core of how the NGBR system was developed. Cytocentric, meaning an approach that takes into account the needs of the cells as they grow in vitro to better mimic the natural environment. This would include accounting for the physiological conditions, extracellular matrix needs, as well as accounting for the dynamic environment and changes in metabolic activity that would naturally be occurring in vivo. The NGBR system takes this into account at many steps along the way. First is the CAD software used to design the cell patterns, allowing users to customize those patterns as they deem necessary in a layer by layer architecture to ensure that the cells experience a more natural environment. Secondly, laser-assisted bioprinting is used to bring those cell patterns into fruition with precision, resolution, and most importantly, the increased cell viability due to reduced shear stress being placed on the cells themselves. Antoine will go into more detail about the benefits of laser-assisted bioprinting in a few slides. So look forward to a little bit more information there. Next, we have the robotic arm and the full automation of the system, which allows for very reproducible results once the fabrication process has been defined. Finally, there's that integrated microscope that we've been speaking about, which allows the end users to be able to observe the precision and reproducibility at each step, but can also be used to monitor the progression of the cells through the maturation process, which is something that Mikhail will speak a little bit more later on in the presentation. As we watch this video of the NGBR system in action, let's talk a little bit more about the advantages of the system. The system was designed with reproducibility and automation in mind for the operator independent fabrication fabrication to ensure reproducible workflows. The integration of more conventional bioprinting techniques along with the laser-assisted techniques allows users to a full complement of techniques to help achieve the fabrication needs to print complex tissue models. At the core of the NGBR system is the precision, accuracy, and reproducibility of the cell patterns. Inclusive in this is the resolution in terms of both volume as well as spatial distribution, which is possible using the laser-assisted technique. And this is all necessary to achieve the cytocentric 4D bioprinting approach. I'll now turn things over to Antoine and then later to Mikhail to help expand on some of the topics that I've touched on during the introduction to the NGBR system. Antoine, over to you.
Yeah, hello, and thanks a lot for inviting us. I must say that both Mikhail and myself are really happy to join you for this webinar. So yeah, my name is Antoine Jacot. I'm a sales manager at Poietis. So I've joined the company back in November 2019, and my job is to help bring this fantastic technology to the market. And I would like to, say that we're really happy to be partnering up with Cintiq for North America. So today I'm going to focus on this different approach to bioprinting that's actually behind NGB-R. That's clearly disruptive and extremely cytocentric. So we're going to talk about what the workflow looks like with NGB-R, a different way to look at biomaterials that's very specific to Poietis. And we're definitely going to talk about laser-assisted bioprinting and the many advantages that the technology offers. So before we get to that, I'd like to highlight the fact that there's not one, but actually three technologies with NGB-R. And the first being bioextrusion. Now, this is probably the most popular and well-known technology out there. This is what more than 75% of people are using. So nothing really new here. It's basically technology that allows you to print a bioink through a nozzle in form of filaments in XYZ, and it's pretty convenient for more viscous gel type biomaterials. So this technology is of course available with NGB-R so that people can keep their existing protocol when moving on to NGB-R, and then incorporate, laser bioprinting in the mix later on. Second is inkjet bioprinting, also called microvalve. It's also a nozzle-based technology, but here we're not printing filaments, but drops. So again, drops of hydrogels or biomaterials, and it's pretty cool for a bit more stable and liquid type biomaterials. And at the very core of NGB-R is, of course, the laser-assisted technology that we've developed. And the main difference here is that's a nozzle-free technology. So cells biomaterials don't go through a nozzle. And I'll explain in a few slides from now how this actually works. So if you look at both nozzle-based technology, you see that you can print both cell-free hydrogels as well as bioink. This is something that pretty much everybody knows, but where laser is very different is the fact that in addition to cell-free hydrogels, you can print cells alone. So you don't need to mix them up in a bioink. Cells can actually be printed directly in their own culture media, and that's a huge difference from what people are actually familiar with. So let's look at that because this is really interesting. Now, let's look at the main difference between conventional 3D bioprinting and this disruptive approach. So with conventional bioprinting, people are usually relying on what we call a bioink. That's a mix of living cells and one or several biomaterials that is extruded out of a nozzle in XYZ. With Poietis, cells and biomaterials are actually printed separately. And the reason is we print living cells directly in their cultural media with the laser head, and that's what we call a bioink. So here there's definitely a definition to be aware of. And biomaterials are usually printed through nozzle-based technology, maybe extrusion or inkjet. And so you may wonder why we do that. Well, there's a huge difference here in how we regard, apprehend, and see biomaterials. If you look at conventional bioprinting, people have spent months and years in fine-tuning bioinks and choosing a specific biomaterial over another in order to minimize negative impact of shear stress, in order to prevent cells from dying, differentiating, or getting injured throughout the process, and sometimes help with clogging problems or cell density when working with high cell concentration. So basically, to sum up, with conventional bioprinting, a biomaterial is chosen based on how to minimize something negative. Whereas with Poietis, it's just here to maximize tissue maturation. So basically, the reason why we're going to choose a biomaterial hydrogel over another is just whether it's going to help induce biological functions afterwards. So it's clearly more biology oriented instead of chemistry oriented, if I may say. So let's look at laser bioprinting in more details. The technology here is nozzle-free. It's also a scaffold-free technology that's able to print cells in a very high density if required, and that's a clear advantage. So here, cells they don't go through a nozzle. They're basically transferred from a donor that we can also call a metallized substrate to a receiver through laser energy. So we're using pulsed laser beams generating enough pressure to create a jet of tiny droplets that contain cells. So if we look at the specifications here, it's pretty interesting because, as you can tell, you can work with many different cell concentrations here, right? So from 1 to 100 million cells per ml, that's not going to be a problem. Again, the printing speed here is really interesting because, as you'll see in the next video, you can print up to 10,000 droplets So these contain cells, by the way, right? So cell loaded droplets, so 10,000 per second. And last but not least, since you can also rely on the laser technology to print biomaterials, the viscosity range here is pretty interesting. Gives you, well, a large choice of compatible biomaterials. So looking at it and more closely, this is how a pattern or a grid, right, of cell droplets gets printed with laser. Pretty fast. And if you look at this through the microscope, well, this is what you get. So the main thing you can take away from this slide is laser lets you distribute cells specifically across the tissue. So if we go back to the entire workflow of a specific tissue biofabrication, this is what it's going to look like. You always start printing a primary bed of biomaterial to ensure that cells will land smoothly, right? You don't want them to burst onto the receiver. And you also want to promote proper cell adherence and adhesion, right? So this primary bed of biomaterial is usually considered ECM, and is printed with a nozzle-based technology for more convenience. Then on top of that, the cell printing starts, right? So we're going to rely on the laser head to print a primary layer of cell droplets. And so from there, all we do basically is just repeat that pattern over and over from a 2D to an actual 3D tissue. And as you can tell below here, the printer just keeps going back and forth from a printing modality to another. And this being automated, well, it ensures a very reproducible workflow. And the last step here is from 3D to 4D, which is something that Mikael will definitely expand on. But basically, this is time, right? So you're going to let the tissue maturate, and this goes to 4D. So if we go back to Tonia's video, here's what it looks like. There's always a first step, which is calibration. Then we crack on with that primary bed of biomaterial using inkjet. And then right before it moves on to the laser bioprinting, it's going to flip upside down since we're printing against gravity. You're going to get a first pattern of cell printed. Then as an option, you can capture an image with the microscope. And finally get to see the receiver. Now, just keep in mind that this is a sequence for just one layer. So if you were to have, say, 18 different layers in your tissue, well, you're just going to loop this and repeat this 17 times more. Now, let's look at laser-assisted bioprinting and its advantages, because this is really fascinating. So there are three main benefits from relying on laser bioprinting compared to other techniques. And we're going to highlight them now. So the first is definitely cell viability. Since cells are printed separately and do not go through a nozzle, they're not suffering from shear stress, right? There's no pressure. So the risk of cells getting injured, damaged, or killed throughout the process is close to inexistent. We've run a lot of tests on so live dead viability tests on the different cell types that we work with, and the average is 93 to 95%. Now, what you see on screen here is with skin cells, so they're pretty easy to work with. But just so you know, we've worked on more than 26 cell types, different cell types with the laser head. And the average was about 93%, which is outstanding. So it works with neurons, neural cells, no problem. And we also have a team currently working on plant cells quite successfully, right? Just so you have an idea. The second advantage is, of course, the control you have, and more specifically, control over resolution, right? So with the laser technology, by playing on a couple of parameters, which Mikael will highlight in a minute, you can adjust the actual number of cells you want per droplet, right? And so this lets you work with both, of course, cellular spots, but you can get very close to single cell bioprinting. I just want to get something really clear here. this is not a pure single cell technology, because this would mean that we're 100% successful every time we attempt to work with single cell. But you get extremely close to single cell, because, well, you can have, from 1 to 3 or 5 cells per droplet by reducing cell concentration. And the last advantage, which is clearly the most interesting here, is the possibility to spatially distribute and organize cells within the bioprinted tissue. Now, once again, if you get to compare this with bioextrusion, there's a huge difference. On the left-hand side, that's cells printed as part of a bioink with bioextrusion. And as you can tell, the only thing you can play on is the actual number of cells in the mix, right? So the cell concentration, but there's no way for the user to control spatial distribution of cells once it's printed. That's not the case with laser bioprinting. And so this time-lapse video of a pattern of primary human fibroblast illustrates this. A pattern is going to have a huge impact onto what happens next. And so this is what we call 4D bioprinting. And this is what Michael Garcia is going to introduce right now. And this is really, really fascinating.
Hi all, so I'm Michael, Business Application Manager here at Poietis. So part of my job is to understand customers' needs and then lead related applicative projects. Other part is to manage R&D projects with industrials and also with academics through European projects. So today I wanted to discuss the notion of 4D bioprinting. So now a lot of people talk about 4D, and I wanted to illustrate why this notion is so important. So when you do 3D bioprinting, you basically organize matrix and cells in space. But what you obtain after printing will evolve along time. Cells proliferate, they migrate, they interact with the surrounding environment. And this environment, so the matrix, will also change, and it can deform, shrink, contract. So that's what happened during morphogenesis, basically. What is important is the initial cell pattern, chemical gradients, and mechanical gradients also. They all affect tissue fate. Here, we will focus on cell patterning, and I will try to show you how you can do 4D using high-resolution laser printing on our platform. So here you have a pattern designed with our CAD. And what I wanted to show you is that with the laser printing, you can control two important things. The organization of the pattern with a resolution of tens of microns. So basically, you precisely print droplets containing cells. And also, you control the quantity of cells inside these droplets. To do that, you play with the laser energy and with the bioink cell concentration. So playing on these two parameters, you can control how much cells you have in your patterns. But now, how can this be useful for you? So we did the first experiment, printing a pattern of fibroblast over a collagen layer. The pattern is the same on the two samples. What differs is the quantity of cells by pattern. On the left, there is a higher quantity of cells. We printed around 50 cells per droplets, and on the right, around 20. What we saw is that at the final, the result is the same, meaning that cells migrate in an homogeneous way. But what differs is the chronology. You see that on the left, you have less cells and they migrate faster. So only playing with local cell concentration enabled to slow down or accelerate some events like cell migration. And this can have a major impact on tissue engineering and in morphogenesis. Then we did a second experiment. So the same material, but this time the quantity of cells by droplets was the same for all conditions. Here we played on the pattern design. On the spacing between the droplets we printed. So you can see here the analysis in light sheet microscopy, and I would like to thank Imactive 3D who provided us these images analysis with their really amazing technology. On the left, we see the three different patterns post-printing, and on the right, we see the evolution at day 2. So again, the result is that cell migration can be modulated, and here just playing on the position of the patterns. So in these two examples, what we modulate is only the chronology, but we try to play at the same time on both, meaning on the pattern spacing and on the quantity of cells by patterns. And here is the result. So here you have the initial pattern. Then at day 1, we can see the beginning of cell migration. And finally, at day 4, we see the final results. So with a precise control by laser of these parameters, we were able to control cell alignment. And here you can see fibroblasts perfectly aligned perpendicular to the initial pattern at day 4. So to summarize, using laser technology, we can precisely control organization of cells. And from this initial pattern, specific biological function can emerge. And that's why resolution is so important to address tissue morphogenesis. Using a similar methodology, we developed a few years ago the first commercial bioprinted skin model, Poietis. Here you have the full workflow. The first step is the design using our in-house CAD. Then you have the fabrication, and the major components of the dermis are fibroblast and collagen. We printed them layer by layer. So collagen is printed homogeneously, whereas fibroblasts are printed with a specific pattern to control their migration during dermis maturation. At day 5, keratinocytes are printed with a different pattern, and I won't go too much into details. I will do that for the next webinar. But here again, there is a maturation step. At each step of the biofabrication workflow, we can assess that the design is respected using the microscope inside our platform. And we do it basically layer by layer. So here we print a layer of fibroblasts, then collagen, and again fibroblasts. We can see the precision, and we can also do some quantification from these pictures. And after printing, we fix the samples at different steps to assess cells' viability, integrity. So here we looked at cell proliferation, adhesion, protein synthesis. We check that everything is going well. And here is the result. So this nice histological section on the right with homogeneous and well-oriented basal layer, presence of granular cells and of the stratum corneum. And on the left, immunostaining of the sample, assessing expression of epidermal differentiation marker, cytokeratin 5 and 10 at proper location. So just to conclude, I wanted to give you an overview of our expertise, showing some projects we have been or are still involved in. So 2 really amazing European projects which I'm leading. ASCTN, which is focused on bioprinting brain organoids to model neuronal disorders, and then Pan3DP. This one is addressing pancreatic models. Then we also worked with Servier on liver models, with L'Oreal, a really great project on hair follicle, and I hope that I will show you some results really soon. And then BASF, so with them we worked on customized bioprinted skin model. You can see the video down. So it's light sheet microscopy, really great video. And Tona, back to you.
Great. Thanks, Mikhail. Sorry, I had a hard time finding my unmute button. So thank you both, Micah and Antoine, for the really great discussion thus far about 4D bioprinting and about the more cytocentric approach of the NGB-R system. I did want to take one more minute of everyone's time just to launch our last poll. And we'd like to understand if it makes sense for us to reach out and find some time to discuss in more detail the work that you're doing or the research that you're doing, and as it relates to the NGBR system that we talked about today. Do you have any short-term plans that you'd like to learn more about the system, or are they a bit more long-term? Or would you simply like us to send you the link to the webinar so that you're able to review it yourself again and share it with any colleagues? Okay, I'll give everyone just a few more seconds to respond to those questions. And again, anyone that has questions, feel free to put them into the Q&A dialog box at the bottom. We have some great questions coming up in the next slide or two. And thank you everyone for your active participation in the polls as well as in the Q&A. We really appreciate getting some feedback from everyone, but also understanding the questions that you have based on the information that was presented today. So again, in summary, I started by introducing the two companies, Synteca and Poietis, and the partnership that we formed together this year. We then took a brief overview of the NGB-R system, and then Antoine dove a little bit deeper into that disruptive technology, as well as taking a look at the cytocentric approach. And then with Mikael, we did a little more look at the advanced cell patterning, the importance of it, as well as the idea of 4D bioprinting. And at this point, I'd like to enter into the Q&A session. And again, I'll work with the team to go through the questions that have come through and we'll provide answers. Any that we don't get answers out to, we will put together a transcript and send that out to everyone who registered for the event today as well. And we hope to get that out to everyone within the next week or so. So to start, what cell concentration do you have to work with using the laser bioprinting system? And perhaps, Mika, you would like to start answering this one, and then if Antoine, you have something else to add, happy to open it up there as well.
Sorry, so it really depends on which kind of tissue you're working on. So usually in our bioink, we use between 10 millions per ml, between 10 millions and 60 millions per ml of cells. And it's with this kind of concentration that we can play on the quantity of cells that we got, that we print.
Perfect. And is there any limitation in the size of the tissue that's being printed? Antoine, perhaps you want to address that question.
Yeah, that's a very interesting question that we get very often. Basically, the only limit is the receiver that you're using, right? So at the moment, NGB-R is compatible with 6 well plates, 12 well plates, and one well plate. If you take the 6 well plate, which is definitely one of the largest, I'd say the max tissue footprint that you can bioprint would be around 2 to 3 square centimeters. We've got a team also working, because as you know, we've got clinical endeavors as well. So we've got a team working on a system to make it possible to print up to 40 square centimeters tissues. So technologically speaking, that's definitely possible. At the moment, it really depends on, you know, what receiver you're dealing with, though.
Perfect. Antoine, another question for you. How much time do you need to print a pattern of cells?
Yeah, that's also a good one. So as you've probably seen with the laser video, printing a single layer of cell droplets, I mean, it's literally less than a second. It's I mean, fast doesn't even cut it. So usually you're not going to print more than 10,000 droplets, so it's definitely going to be less than a second. Now, just keep in mind that you may not be printing just one single layer, right? Usually when you want to produce a tissue, it's going to have several of them. So you just have to multiply that time by the number of layers that you wish to print. And the other thing that you need to keep in mind is that since you're going to be printing layers of biomaterials with nozzle-based printing technology, that's also some time that you need to account for. So again, as Mika said before, it's really hard to give you like an example, because of course it depends on literally how many layers you want to print and what you know, printing modalities you're relying on. But generally speaking, I don't know, it could be from 5 to 20 minutes.
Perfect. And we saw definitely in the video that you showed of the laser-assisted bioprinting, how fast that pattern of cells could be placed onto the well. It was, to me, quite fascinating and something that I really enjoyed watching. Definitely. Micah, why is the microscope built in rather than using an external microscope?
So yeah, a really good question also. So as an on-board device, the microscope is really integrated right into the NGB-R workflow. And this is really convenient. I mean, when you do experiments, it's always complicated to go outside and then to go back on the bioprinter, then you move the sample. This is not so good. There is difference in temperature. So it's really convenient to get it inside your machine. And I mean, the way of working, it's a manufacturing process, meaning that you launch a sequence and then you don't want to stop it to just register some results. So that's really convenient to get it inside. All is recorded inside the machine, and then you can go back on that just after your experiment. So I mean, it's convenient, really.
That makes sense. And perhaps, Antoine, we can address a question regarding the fluorescence capabilities. Does the microscope have fluorescence capabilities, or even adding on to that confocal capabilities?
Yeah, no problem. So yeah, the microscope remains an option. That being said, it's an option that many people really want to have included with their NGB-R platform. And the main reason is it's not only a QC, so quality control tool, as Mika demonstrated before, right? So capturing an image after every single layer was printed. It's more than that, because you can use it to do what we call targeted bioprinting. So you can pre-visualize the receiver and then at the touch of your fingertip, you could basically select the areas on which that you'd like to print cells and do just that, which is extremely cool. Because I mean, then you stop just thinking in terms of 6 and 12 well-played receivers. You start thinking about incorporating cells onto chips, like or special devices, and even 3D objects, right? But yeah, to answer your question, at the moment, the microscope is not a fluorescent one. But I don't think that it would be a problem if we ever had a customer for whom that's a requirement.
Fair enough. I guess a little expansion on that. Is it possible to program the robotic arm to go to a specific place within the cabinet if an individual is to place something inside that cabinet?
Oh yeah, and that's why having onboard imaging devices comes at a huge benefit, because that's exactly what it can do. So you can use those either the microscope or the onboard wide field camera. You can use those devices to see where you'd like to incorporate cells at a with 2 microns precision. And then just with the laser technology, basically choose what kind of cell concentration, or at least a certain number of cells you wish to print at this specific spot.
Great. And then one last question, not last question for Antoine, but a question that we do have for Antoine is there an integrated incubator inside the system?
No, there isn't. Surprisingly, a lot of people are usually asking this question because I guess the reason I explain it is when you look at it from the outside, it looks like since it's it's a real platform. It would make sense. To be honest, a couple years ago, I know that the team was considering it, but one, that would add up in terms of cost. The instrument is quite premium already, so we really didn't want to make it more expensive than it is. Second, there would be a space problem. So as you can see, since it's an actual biosafety cabinet, the footprint is pretty large, right? So we didn't want to make it any larger. And then 3, since the actual bioprinting process is quite quick, that means that your tissues can be taken back to an incubator pretty quickly. So there's not like a crucial need for an onboard incubator anyway.
That makes sense. Talking about temperature and incubators, is the biosafety cabinet kept at a specific temperature, or are we printing at ambient temperatures?
Yeah, so we print at ambient temperature. But I've seen a question in the Q&R section about thermoregulated printing heads.
Yes.
And so, yes, printing heads can be thermoregulated individually, of course. And as you've probably seen in the picture, there is room for 3 nozzle-based printing heads. So again, just for those who weren't with us, what we call nozzle-based bioprinting heads are either bioextrusion or inkjet bioprinting. And so those can definitely be thermoregulated should you need them to, depending on what biomaterials you work with. And temperature can be controlled live through the software that's already installed on the machine. And of course, that temperature control is individual for every single one of those.
Perfect. Micah, this one I think relates back to you. So you talked about the full thickness skin model and how 4D bioprinting relates to that. Can you just expand a little bit on which types of cells were used in that model? For example, were neurons, adipocytes, as well as immune cells used for that model, or how was it built up?
So regarding this model, we used fibroblast and collagen for the dermis compartment, and then keratinocytes for epidermis. So we didn't complexify it much more. Then we've got others project. I can't communicate on that. In which we try to complexify, yes, our model. It's not with neurons, but I can communicate. Sorry, more about
For
that.
sure. With, yeah,
yes,
I'm sure.
We
Yeah.
printed other cell types. Yes, of course.
Perfect. Okay. Yeah. I'm sure there's NDAs with different groups that the team at Poietis is working with that's preventing. No, it's okay. I fully understand. From the same person asking the question, how do you usually optimize the cell culture medium, and how do you go about really optimizing the whole fabrication process? There's obviously different components that need to be optimized. Can you talk to us a little bit about that process?
So I'm unsure to understand the question. So it's related to the medium that we use. So not on the workflow process.
Not on the workflow process, but how do you optimize the culture media that's used, I guess, to have the best cell viability, but then also probably the bioextrusion of the extracellular matrix components as well?
It's a really tough question. It really depends on your samples. And there is 2 things. There is, we always assess something outside the machine, and then we assess the process of the machine. So these are 2 different things, meaning that we can control things because of the machine, so the pattern, etc. And this we assess that there is, it doesn't affect the cells. But then regarding the effect of the medium, we tested before all of this. There is a part of the experiment where we assess all the material that you got. Here it's really complicated to respond to this question without the purpose. I mean,
sure.
If it's on a specific tissue, I can address it, but like that, it's complicated.
Absolutely. I will reach out and try to get a little bit more information, and we can address the question offline. That's no problem. Yeah, okay. Of course. I'm just looking through some of the questions that came through, and I'm trying to actually link the questions together. So just bear with me for a second. A super, maybe not a super easy one, but a really quick one is, how fast do the droplets, which are shot out by the laser-assisted bioprinting, how fast do they travel through the air? Do we know?
Okay, well, I guess that depends
really fast,
on,
but I don't know.
It's a cool question. It means that people are interested in the technology itself, and I'm very happy to address that. I guess it really depends on how far out the receiver is from the laser donor. But generally speaking, it's about a couple millimeters only. So in terms of timing, I mean, it's microseconds, even less than that.
Perfect. Related a little bit to that in terms of the laser-assisted bioprinting, as you prepare the laser-assisted bioprinting, you take your cell media and you place it on the metallized substrate. How long do the cells stay on that metallized substrate before evaporation takes place and perhaps risk the cells dying? And do you have to replenish that source over time?
Tonya, Sorry, Tonya,
Yeah.
I just wanted to add something to
Sure,
what
please.
Antoine said before. Yes, because you're speaking about the velocity of the droplets. So of course, when we print, what is important is to print on a soft materials to prevent any issue on cell viability and in cell integrity. So I don't know if the question was related to that, but yes, if I have to pinpoint something, is that when we print with the laser, we print on a soft surface.
Got it. That makes sense. And it may have been related to that based on this second question that I posed comes from the same person, taking a look at how that evaporation of the media on the metallized substrate for the laser bioprinting, how long you have until you need to replenish that, or if there's any effective evaporation within the fabrication process.
That's a good question. Yes, the point is that the printing process is really fast. So usually the donor doesn't stay that much time inside the cabinet. But yes, if you have some evaporation of the medium, you can then modulate the laser intensity of your laser. So according to the protocol that you have, at some point, if you don't want to use all the ink that you've got on your donor, and you have some pause, the next layer that you will print, you can adapt by reducing the laser intensity. So I mean, there are different ways to play with that, but yeah, if you wait like 20 minutes, you will have a lot of evaporation. So I would say to prevent evaporation, it's you have to print in less than 5 minutes, and then you just have to adapt to that.
That makes sense. Great. And question here: how do you print and cross-link multiple layers of collagen? So I think one thing that we didn't talk about, and perhaps I'll take a stab at answering this and open it up to Antoine or Micah at the end. We didn't talk about other options available on the system. We talked about the integrated microscope, and the wide field microscope is also an option, and Antoine has brought it up. One of the other options is to have a UV crosslinker built into the system, and then the arm would simply bring whatever sample you were printing. Underneath, there's multiple wavelengths. I think it's 305 and something, 400 something nanometers to provide the cross-linking for the collagen, and then you would continue on, and that would simply just be a step in the fabrication process. Antoine or Micah, anything else to add to that one?
So regarding the collagen, you've got different ways to gellify it. In our case, we print like, I think it's 5 or 6, it depends on our models. But to do jellification, we take outside the samples to the incubator in our case to make this jellification. So the process is longer, not because of the machine, but because of this jellification time.
Perfect. An interesting question, and Mikhail, it's probably going to be you that will start with the answer. How do you start to figure out the pattern in which your cells will thrive the best? And is it just simply a trial and error, or relating to experience? And perhaps what could Poietis's role be in that as it relates to new customers that are starting out with the system?
So really good question, and that's precisely the point. At the beginning is yes, we try. I mean, we got many errors, and at some point we gain some knowledge, and previous results can be really useful. And as we are addressing a lot of kind of tissues, a lot of kind of cells here, at some point we've got some expertise, and we were able to know at the very beginning which kind of pattern we need to use. But one thing I can say is that at the really beginning, what we did is really to observe what happened during cell growth inside the flask. So here you really see how cell behave, and what we try to do is always to reproduce what do cells, but we just accelerate it, we facilitate it, let's say.
Perfect.
Yeah. Just want to add something because this is a cool question. The only thing I want to say, and that's the whole point of the NGB-R, right? So this is what the technology is offering, and it's precisely the point of investing into NGB-R is for people to be able to find out, right, and work exactly on this.
Perfect. Great. And I know the team has a tremendous amount of experience, and I think in the future it may be really great to create kind of a user network in which people can exchange ideas, because there's no need to reinvent the wheel, especially in basic research and academia, to be able to help one another and build that community amongst those kind of doing the same type of work.
Yeah, and you know, Tanya, like as far as I'm concerned, the reason why I enjoy working at Poietis so much is I know I've got a team behind me, and behind, you know, the whole company, a team of biologists, Mikael and other people that are actually working on the instrument. So we're not only manufacturing things, it's an instrument that was designed by biologists and for biologists. And the team we have is definitely willing to support any customer with, you know, laser settings and parameters, and share the experience to basically, you know, save time, right?
Yeah, no, that makes sense. An interesting question, and either of you guys feel free to jump in. Can and has the technology been used to print a network of microvessels? So this is something that Antoine and I have talked about a bit in terms of how do we start to get functional vessels within the tissues? And so has it been used to create a network of microvessels, which will obviously depend on different cell patterns and types? And do you think it could also in the future be scaled up to print organs?
So actually, we never worked on vascularization. We will maybe do that because we are writing some project related to that. I know that there are different approaches to do that. There are people using extrusion to print sacrificial layers and then add some cells inside through microfluidic. Then there are the guys who are printing the cells with the laser, and they are playing on a certain ratio between endothelial cell and mesenchymal cell to obtain through cell autogranulation to obtain these kind of vessels. So I mean, there are different techniques. What is nice with our bioprinter, with our platform, is that we can combine all these techniques. So it's quite good for that, I mean. So we could do that, but yes, actually we didn't do that.
Great. As we approach the top of the hour, perhaps I'll pull together a few different questions relating to the idea of single cell bioprinting. And Antoine, you and I have talked about this a little bit, but how does the system, and can the system truly do a single cell, or is it more a matter of laser intensity and concentration within the droplet with the potential of getting down to a single cell? And obviously then, can the system identify the cells within the metallized substrate and select that cell, or is it simply the concentration, again, the energy of that laser beam and where it hits the metallized substrate that would affect the ability to print a single cell?
Yeah, that's an outstanding question. There's a few things to say to that. So at the moment, there are basically two parameters that you can play with, and this is what Mikael described before. That's of course the amount of cells, the quantity of cells, cell concentration in the bioink that is spread over the laser donor. That's something you can play with. So of course, if you want to get close to single cell, you're going to have to reduce that cell concentration. And the other parameter that you can adjust is how powerful the laser beams are. And once again, if you want to have like smaller drops, you're going to have to reduce that intensity. Now, it's not, how am I going to say this? It's not 100% accurate, as you can imagine, right? Because all you have to do is play on those parameters, but you can get very close to single cell, but it's not going to be 100% of the time successful, meaning that if you want to print, say, 100 droplets and you only want to have one cell per droplet, you're not going to have them all, right? Like some of them
Yeah.
are definitely going to have one cell, some others are going to have a couple of them, maybe a few drops will have 3 cells, and maybe some drops will be empty. And this is something I really want to emphasize on, because it would be wrong to say that we have a single cell technology. What we have is a technology that's getting very close to single cell. And that's extremely convenient because you have an amazing control over resolution. And the other thing I want to say is we've got a part of our team working on, so it's not a feature that's available now, and it's definitely not going to be available in the short term. But we're thinking about having like a camera located right underneath the laser donor, so that you can visualize cells directly on the laser head before they get ejected. And so this way we would get very close to single cell.
Right. That makes perfect sense. A
By
number of
the
other
way,
questions. Go ahead.
Yeah,
Sorry, go ahead.
yeah, sorry, Tanya. I just wanted to add something. What I've described, so having this, camera device located underneath the laser donor that lets you visualize the bioink, we've actually used that for organoids. So I don't know if you've remembered one of my slides where I commented that it can also work with cell spheroids and aggregates. That's a way you can do it. So the idea would be for you to visualize exactly where the aggregates are located across the donor, and then ask the technology to shoot exactly at that location, right?
Right. That makes perfect sense. Excellent. We do have lots of other questions. I will work with Antoine and Micah to get answers to those. And again, we'll send them out as a transcript in hopefully within a week or so to everyone. But we have reached the 60-minute mark, and I thank you everyone for your participation. It's been a great discussion following the presentation. And we do want to be respectful of everyone's time, so we will wrap things up at this point. I'd like to thank Antoine and Micah for joining me today as presenters in this webinar and taking the time to address as many questions as we did of yours, and their future time in helping me to answer the rest of the questions. I do trust that we've been able to provide you with some exciting information about the potentials of 4D bioprinting and the laser-assisted techniques using the NGB-R. If in the days and weeks to come you have further questions about the system that we talked about today, I do encourage you to reach out to myself at Syntica. And we'll be happy to discuss further, and I can pull Antoine and Micah in as needed. I would welcome the opportunity. It is a new area of research for me, so I would actually welcome the opportunity just to talk about your science and what you're doing and how you're using 4D bioprinting, just to help me learn further. And again, I want to thank everyone for taking the time to join us today. Again, your active participation in the webinar makes everything so much more fun for all of us doing the presenting. And we do look forward to either speaking with you in the future or seeing you at another Syntica instrumentation event. Thanks again, and have a great day.
Great job, everyone. I mean, we're still on, I
Hey,
guess.
yeah, how did it go?